First of all, I wanted to begin by thanking the organizers for giving me this wonderful opportunity to come and hear from the experts. And then, before I actually begin the talk, I wanted to tell you a story. The story is this: that last November, myself and Anton and Peter and a few others found ourselves in a mansion somewhere in UK. Now, UK is not is a supposedly a rich country, but mostly made of not so rich people. So where that richness is another story. But but anyway, we were there, and this was a conference, and so where there were lots of biologists, and I felt that I was an imposter there. So I kind of said at the beginning of the talk that, this is a talk where, there are some talks in a conference where you feel that maybe if I skip it or if I sleep through it, then nothing much will happen. And then after a few months, I got an invitation from Anton. So I think he must have followed that advice very carefully. Right. Okay. So what we are going to talk about today is I'll tell you about what is surface migration, why is it important, and how do you control it. We'll give our perspective. And then this will nicely segue into what is the role of elastic interactions in the formation and functioning of membraneless organelles in cells. This is in line with what people have been talking about, not only in terms of chromatin, but almost every piece of biological literature that is coming out saying everything is biological phase separation that is going on. Whether it's true or not, time will tell. And then if there is time, then I will tell you about epigenetics, chromatin, lamin, and all that, and what I believe is our hypothesis as what the large-scale organization of genome might be looking at. So I begin by providing the acknowledgements. All of this was hugely motivated by my mentor, TCB McLeish, when I moved to Durham several years ago. All the theoretical work that you see here are done by a few talented postdocs and PhD students, Biswa, Shubho, and William. The simulations have been done by us, but overlooked by Mark Wilson, and the experiments were done by Richard Thompson and Jason Zhang. And these are the people who provided funding with almost an equal split between research councils and industries. Okay, so in terms of time scales and length scales, this is a plot that I like to show where the bottom corner is the world of very small and very fast, and the top right corner is the world of very slow and very large. And we are somewhere in this happy domain where we use coarse-grained molecular dynamics simulations, some mesoscale hydrodynamics by the way of time-dependent Ginzburg-Landau kind of theories, and then we also write down pen and paper theories. So with that introduction, let me tell you what is surface migration. So if you have a mixture of polymers, let's say two types of polymers, let's make our life simple. One which is this red chain, long chain, wiggly ones, and the short black ones with a surface that is exposed to the atmosphere. Then as time goes on in equilibrium, you will see that there is a increase in concentration of the little guys close to the surface. And then you start thinking about why this happens. Well, maybe what is happening is that when the polymers go to the surface, then they lose some entropy because the configurational space available to them is less. If it's a long chain, there is a more decrease in the number of degrees of freedom. But then you, of course, gain some surface energy by associating to the surface because you don't have so many bonds with your neighbors as opposed to when you're in the top. And so the balance between these two will dictate how much of material will migrate to the surface. So if you are looking at the surface fraction as a function of depth, assume, for example, that there is two-dimensional homogeneity. I'm just looking at it in the depth. Then I should see this profile like the blue line, almost like an exponential decay, where it starts from a large value, and then it decays to some constant value in the bulk. Now that is, of course, assuming that A and B like each other, the red and black like each other, and so the bulk of equilibrium phase is a homogeneous mixed phase. If, on the other hand, they don't like each other and it undergoes phase separation, then you're going to find that all these black molecules come to the surface, and so you essentially form what is a macroscopically thick wetting layer. So in that case, the profile looks like that red line where you have a large black layer, and then it somehow, over some length scale, decays to the bulk. Depending on how large that length scale is, your interface will be sharp or diffuse. So that's the picture that we have. And of course, this is not a theorist's imagination. It has been experimentally verified. So you shine some X-rays onto a block of material, which is a mixture, and then this have a finite penetration depth. So if you tilt the material with respect to the incoming X-rays, then the more the angle changes, you're going to probe molecules closer to the surface. So if theta is 0, which is basically you're shining directly on top of it, then this polystyrene and PVME is polyvinyl methyl ether. They have almost nearly equal distributions. But look, when it is large, that is you're grazing the surface, then one of these has got a large peak in comparison. So if our ideas are all right, then we should see that there is a difference in the surface energies between these two polymers. And of course, you can do this simply by putting it into a tube and looking at the drop shape. You know that a liquid drop will have a certain shape. You can fit that shape with a single parameter, namely the surface tension, and then you can find that out that there is indeed a difference. The gamma of PVME is lower than gamma of polystyrene. So all of this was kind of known. And so if this idea is working, then if we try to make a theory, then this should have two ingredients. The first thing should be the thermodynamics of polymer solutions, which will tell us whether the molecules like to be mixed together. And then I combine that with the wetting of a wall, wetting of a fluid by the wall, in this case atmosphere. And our aim is to predict whether what kind of a variation of this fraction as a function of depth we should get, whether we should get this kind of a non-wetting profile or whether we can get this wetting profile. And of course, a single parameter can change it, which is the chi parameter, which is how the two molecules like each other. Now, I grew up reading books that Shura and others wrote, and the simplest model that you can write down for a polymer is Flory-Huggins theory, where you say that I have got two chains, two types of molecules, molecular weight Na and Nb. There is a term which is proportional to which is the entropic term, and then there is an enthalpic term that depends on the association of the A and B type of molecules with a constant or the miscibility parameter that is inversely dependent on temperature. So therefore, if I increase my chi, that means lowering the temperature and vice versa, and you can estimate that this chi is somehow related to the difference between the AB interaction, difference of AB interactions and the self interactions AA and BB. So okay, for some parameters, you can put it in MATLAB or Mathematica and you can plot it and you get a single minima, which means that the ground state is homogeneous or mixed, or in other cases where you jack up the chi, then you get this kind of a profile where the system has two minima corresponding to domains which are A-rich and B-rich. Now, the way you can work out the thermodynamics of this is to say, well, I'm going to draw a common tangent to these two minima, which essentially ensures that there is the chemical potentials are the same. So there is no mass transport that is taking place across these domains. And also the intercept is actually related to the negative of the osmotic pressure, so that these two domains are in kind of mechanical equilibrium with each other. So this construction, mainly the common tangent construction, will essentially ensure that you are in equilibrium, and the phase where you go from a homogeneous to a demixed phase will look something like this, where you have now changed to red and green instead of, that was a bad thing, should have kept red and black, but there you go. Okay, so that's how you understand one ingredient. What about the other ingredient? Well, essentially the wetting of a fluid. If you take a drop and put it on a surface, then in most cases you will get a hemispherical drop. and then you can do a balance of the interfacial forces that are acting between the liquid vapor, solid liquid, and solid vapor. And so if the tensions pulling the drop inward is more than the tension pushing the drop outward, you're going to stabilize a partially wetted phase. And if it's the other way around, then it's going to spread thinly to form of a liquid. Of course, these interfacial tensions are functions of temperature, so therefore you can go from a completely wetting phase, where the system completely wets in the form of a surface of a uniform layer, to the being partially wetted or in the form of drops. Okay, you can set up a Landau theory in this case. So if you have an interface between a liquid and a vapor, then these would correspond to the two minima, the equilibrium minima that you had, that were coexisting. And then there is a variation of the density as you go from one to the other. So you can write this interfacial energy in terms of this bimodal free energy density, plus some term that takes into account this kind of variation in the density. You can do this maths, it's all standard classical mechanics. You can do the variational calculation. You can find out from here how z varies as a function of phi, and then turn the plot on itself, and you can get phi as a function of z. So I'm going to skip this detail, it's again very standard, and eventually you get this kind of a profile where you start from one of the minima and you go to the other minima with over a certain length scale. Now that was when you had just two, you were looking at the bulk property. What happens if in addition you have this in contact with a surface? Well, there might be a preference of a particular molecule close to the surface, which can be A or B, and that's going to be proportional to the local density that is there at the surface. So in addition to this free energy that you just minimized, you will have a term that will tell you that there is a preference of a particular molecule to the surface. The simplest form that you can take is that, okay, I associate an energy with a minus, so it'll take a polynomial form. Let's not invent the wheel. It's basically something like minus a linear term, which is proportional to phi s, and then the quadratic term with an opposite sign. The idea is that I put a molecule to the surface, it lowers the energy, so therefore there is a propensity of going there. But once I have put one molecule, if I then add another molecule there, then it costs extra energy, this repels, and so therefore that is the potential form that you use. Again, this is all standard. You can then now do the full equilibrium calculation. You have a part that is proportional to this extra free energy, the variation of that with respect to the volume fraction, and you couple that with your bulk phase, and you get your wetting phase diagram. So therefore, you start from a value at the boundary at z equal to 0, that is proportional to this phi s, which is the volume fraction at the interface. You go from there to the equilibrium value of one of the phases, and then eventually, unless after you exhaust the entire material, you will flow over to the other. So all this was known. We all did that. We found the calculation. It was fairly 1970s. And then we were stumped by this problem from a company that makes nappies. And essentially they said, yeah, it's true, that, well, after a while, these nappies have adhesive patches that they lose their adhesiveness. And this is because functional molecules have migrated to the surface. These are tachyfiers. The same problem happens in chocolate, for example, where the fat blooms out of the matrix. The same problem happens in paints, and I have a list that I can show you that this cuts across numerous industries and products. Okay, so the idea is that they came to us and said, "Can you put us a shelf life on our products?" And we said, "We can't do that, but we can tell you how you can control it." And the idea is very simple. You take the polymers with long chain molecules which were floating in solution, and you start stitching it. Now, therefore you get, as opposed to two polymer mixtures, you get a small molecule that is phase separating maybe and then growing in a matrix that is a gel, okay, a cross-linked gel. So if the droplets are very small, then nothing happens. It does not see the gel network. But once it starts growing and hitting the average size of the gel, then if it order to grow further, it has to deform the network. Now, if you go back and to your original understanding of free energies, and then you think of the background network, you see that the number of degrees of freedom that it has is negligible in comparison to the individual droplets or small molecules. So you say, okay, I'm going to neglect the entropy of the network, but then I'm going to have to put in another ingredient, which is the elastic deformation of the network as the molecules are growing. So therefore, you have the Flory-Huggins, which we discussed earlier. Now you don't have the network entropy that you set to 0. Instead, you have an elastic term. We've taken one form. We'll choose a different form. The details don't matter. The numbers change, but now qualitatively the same. And then essentially what you get is, as you increase the modulus of your gel, you go from this wetting profile to back to your segregating profile. So earlier you had one knob, which was the immiscibility parameter. You changed it. You went from a segregating profile to a wetting profile. Now you have another knob, which is the elastic modulus. You turn that and you can go back. So it probably gives you a little bit more control. So the theory of this, which I'm not a very confident man, so I was unsure that we got it all wrong. So therefore, we did several simulations, both coarse-grained as well as mesoscale. So essentially, we have end-linked gels, and in that, we are putting small molecules, and then we watch it as a function of time. Of course, we can do a coarse graining where you divide your system into small boxes, count the number of monomers, use a majority rule. If there are more polymers than monomers, you take phi to be +1, otherwise phi to be -1. In this coarse-grained picture, you have a different framework. You can use the same form of free energy. You can look at the ordering behavior of this associated with addition energy with the wall. And then this is what you get. As you increase your bulk modulus, then you start getting de-wetting, or essentially not surface segregation and wetting on the walls. And essentially, there was a question that Shura asked, I think, in one of the talks, which is, once you have a gel, what does it happen? How does the dynamics change? Well, you can look at the correlation, the domain size coarsening as a function of time. Typically, if these were polymer solutions, you would expect a Cahn-Hilliard or T to the power 1/3 growth, where if you have cross-linked gels, you will get saturation at much earlier values. So I'm going to play this movie, where on the left-hand side, you have oligomers that are phase separating and wetting, in a gel. And on the right-hand side, you have exactly the same system, where the background has been, of course, blacked out in both cases, where the system is a polymer solution. So what you should see on the left is that the number of droplets are a lot more. They are contained in the gel, as opposed to the number on the right, which are much less. And most of them are going to the surface. Essentially, there you'll see material essentially just wetting the surface. Fast forward to biological systems. This has been discovered in context of P granules and a lot of organelles which are there inside cellular organisms, where they exist in membraneless form. And the idea is that they are kept from dissolving in the surrounding fluid by a non-vanishing interfacial tension. So therefore, if you want to do the same thermodynamics calculation, you can use the same machinery. I'm going to skip it, but with a certain caveat. Earlier, you had a system where essentially we're looking at the bulk phase separation, but here, depending on the volume fraction, if it's less than a certain number, you're going to have a small liquid drop that is immersed in the background fluid. Remember, we did a common tangent construction based on which we said that, well, the mechanical forces are the same and the chemical potentials are also same. So there is no mass flow and there is no deformation that is going on here. But now, of course, when you have a curved interface, we know that there is a non-vanishing Laplace pressure that is acting across the interface. So instead, you have to relax your constraint of doing a common tangent construction and do something we call a parallel tangent construction, where the slopes are the same, essentially ensuring that the chemical equilibrium is maintained, and the difference in between the intercepts is giving you the Laplace pressure to gamma by r. We call it parallel tangent construction. Mike Gates calls it uncommon tangent construction. You can choose whatever name you want. Okay. Well, we can do this, and then essentially we create an ansatz where you have these droplets that are immersed in a matrix. You can write down a free energy. We use a different form of free energy. Why not? We use the sort of the, I don't know why it's Neo-Hookean written here. It's not nice. This is basically the Ghent form. And then you can minimize this. Essentially, this is the same ingredients. One part has been blacked out. You now replace it with an elastic free energy, which is slightly more complicated than what we had used. And then eventually you get this result, which I'm going to show here. So on the left-hand side is, on the y-axis, is surface energy, suitably non-dimensionalized. On the x-axis, you have the shear modulus or the bulk modulus of the system, or the elastic modulus of the system, again suitably normalized. And then if you see the following behavior, if your surface energy is high, you essentially see that the equilibrium phase is made of a single droplet, whereas if the shear modulus or the modulus values are high, then you start seeing that it's made of multiple droplets. So in a sense, the elasticity kind of renormalizes the interfacial tension to create or to stabilize a microdroplet phase. You don't have to do all this. So the dotted line is essentially our calculation, and this dashed line is essentially a scaling calculation that you can come up with by purely saying that I'm going to balance the surface energy with the elastic energy. And this is essentially written in this form, 3 gamma divided by 3 R naught. R naught is a typical length scale in the system, which could be the average mesh size. Now, of course, this is the paper not ours. This is a paper by Pierre Ranseret and others that I really like, where you say that, well, we had talked about this scenario where you went from the fibers, which are with the droplets which are dispersed in this micro droplet phase, and then another phase where you have a single droplet and the fibers extruded from within the droplet. But it's also possible that this droplet actually wets the fibers. So essentially, you can come up with another dimensionless number, which will be the ratio of the surface energies between the droplets and the polymers, and then again divided by the bulk energy of the system set. So this is what I wanted to talk about. My KPI is over. Now I want to tell you about some other aspects of the story, which I think might be of interest to this audience. How much time do I have? 3 or 4 minutes? So this is a story that I started with Mithun when he was just coming off his postdoc, and he spent some time with us. And then there was the series of people that he has now trained. And of course, the first calculation, again, I must mention Tom, because essentially when we started working on this area, I went to him with this idea that we had, and he said, you know what, that's a good idea, we should do it. Later on, we found that, yeah, maybe there were other better ideas. But anyway, so this is the model that I want to talk to you about. Well, we have a heteropolymer in a confined environment where it's made of 2 types of molecules, A and B, or red and green, where there is one type that associates with the wall and the other type does not. Okay? Now, if you can set up a Hamiltonian for this system, you can think of these as springs. You can put in self-avoidance if you want. You can put in semi-flexibility if you want. And then there is an association of these monomers to the walls of the containing vessel. And then you can start thinking about what are the important length scales that are there in the problem. The first thing will, of course, be the confinement radius that is actually telling you that this is not free, but it's within a certain volume. The second thing will be the persistence length, which will tell you how much the length scale over which tangent vectors will decorrelate. And then the other important thing that comes directly in play with this is the length scale over which disorder, and by that I mean the red or the green patches, are correlated. So if you think that I have a very flexible chain where there is a certain length scale over which this is disordered, then what's going to happen is that it can crumple and start forming what are called core shell particles, where the inner hydrophobic cores or non-associating cores are shielded from the external ones. However, if the persistence length is now large, then you are not going to get these morphologies. You're going to get braids and so on and so forth. We initially did this without the confining vessel or confining container. And then when we published this, we realized that this is not our story, but this is part of a story that Shura and many other years ago, Alexei Koklov and others had done, where they said, well, I'm looking at a protein condensate problem as saying that I'm just going to paint the nearest neighbors, and then I'm going to stretch it out and look at what are the optimal sequences on the backbone. Okay? So what are the other length energy, competing energy scales that are there in the problem? Well, you have bending energy, you have this AA, BB, and AB interaction energy, which essentially gives you chi, and then you also have the Lamine association energies. By the way, again, this is not us who have done this problem. The two Davides in Edinburgh have worked on this problem quite a bit, along with a lot of others. And so what is it essentially you get? Well, you kind of start getting obvious things. If you have 4 chains, let's say in a confining vessel, where none of them are interacting with the lamine, then you can calculate these contact maps. You can also calculate lamine proximity indices, which you can measure from DAM-ID experiments and so on and so forth, and they kind of agree. And instead, if you have 2 of the 4 associating, then you start seeing these patches and similar things that you can match with experiments. Right. So what is the story that I'm trying to tell you? I can go into the details of this, but maybe I should not. Is that there is this whole idea that people have been putting forward, Geeta Narlikar and her collaborators in San Francisco and other places saying, oh, there is this chromatin that is with the transcription factors. They are undergoing some kind of a liquid-liquid phase separation in the bulk. And then after Yuval's beautiful experiments along with others, which show that, oh, there are the lamines which are also playing a role in the association of this. So if I think of this from a purely naive physics perspective, then there are 2 things that are going on. There is wetting probably that is operative at the lamine, and then there is probably some phase separation and gel elasticity kind of thing that is going on inside the bulk. Maybe these 2 are playing a role in dictating the physical properties of the system. There may be other things, for example, active processes. We have completely ignored this. In this process, what we've talked about are equilibrium thermodynamic effects. Then the other thing is, what about rigid versus soft confinement? I can knock out these proteins, and then I can look at that there is this is like a wobbly jelly-like confinement. What effect are there on this system? And I don't have time to tell you this, but there is another completely different scale at which you can look at things, where you say, well, I'm going to talk about cell-fate decisions and things that the cells are changing, not at the level of microscopics, but thinking in terms of this whole idea, which was posted around 1970s by a bunch of mathematicians, which said that, well, if I look at all the cells developing, they're starting from one common state, and then as they come down the development pathway, they make cell-fate decisions. They go one way or the other. So the differentiated cells can be thought of as all of those at the bottom of the hill, okay? And then there were these 2 people who did the following experiments. They said, okay, let me go the other way around. I want to take these somatic cells and I want to induce pluripotency. So I'm going to put in some factors, thank you, and then I'm going to just wait. So this is what they see. They wait for 8 or 9 days. They withdraw their chemical. The cells go back to where they started. They wait for 14 or 15 days, and then they leave, take out the drive, and they go to commitment to pluripotency. And then they do something. If you wait in between, then nothing happens. It doesn't come here. It doesn't go there. So semantic mischief, they call these cells area 51 cells because no one knows what is going on. So we looked at a very simple model where we said, well, you're putting in chemicals, there are things that are going on there, and then you have these expression levels of genes being described by these kind of differential equations. This is not an instantaneous system. It just doesn't react the moment you put it in. There is a time delay for the uptake of the chemicals and for things that are going on. This could be various things. It could be DNA methylation. It could be other things. We don't know. It's all washed under the rug. So if you take this and you solve this problem, then, or numerically simulate, then this is what you get. For a particular, I'm going to skip it because Anton is here. So if you plot these two time scales which are there, one is the delay time, that is for the uptake and the self-feedback mechanism loop of this network, plus the time that you have the drive on, then you have a series of transitions where you go from somatic to pluripotent. There's nothing sacrosanct about the way you look at it. It can be flipped. But then beyond this point, there is a bifurcation above which you get a series of transitions where you go from one state to this area 51 state to the other, which is marked by a series of oscillations between the two gene expression levels given by these kind of oscillations. Whether this is there in real life or not, I don't know. The picture that we have is in line with this Waddington's landscape, is that you are trying to come down the developmental pathway and you're trying to make your self-aid decisions, but in the meantime, you have actually changed the landscape. So therefore, you are kind of lost and wandering about in this state space of gene expression levels. It's like playing bagatelle, if you have played it ever, where I'm continuously tilting the board so it does not fall in any of the holes. So I think that's all that I had. I'm sorry I took up a little bit more time than was required. These are some of the places where we have published some of our papers. Feel free to read them and tell us where we are lacking and what can be improved. And thank you very much for your attention, and thank you.